Welcome to this lecture on the basics of neuroscience. Today, we are going to explore one of the most fascinating and crucial mechanisms in the human brain, G-protein-coupled receptors, or GPCRs. If you have ever wondered how a tiny drop of a chemical can completely change your mood, wake you up, or help you form a lifelong memory, you are in the right place. We will build our understanding from the ground up, starting with a simple analogy, moving through the biological mechanisms, and finally looking at the mathematics of how these signals are amplified. Let us dive in and uncover how the brain regulates its own complex behavior. To understand how these receptors work, imagine you are standing outside a highly secure bank. You have an urgent message for the vault manager, but you cannot just walk through the doors. Instead, you hand your message to a security guard outside. The guard does not open the door. Rather, he uses his walkie-talkie to call a team of workers inside, who then run around and make the necessary changes. In the brain, a G-protein-coupled receptor works exactly like that security guard. It sits on the surface of a neuron and receives chemical messages from the outside. Instead of letting ions flow straight in, it signals a special team inside the cell to do the heavy lifting. This indirect method allows the neuron to create massive, widespread changes from just a single tiny message. Now that we have our bank analogy in mind, let us look at the formal biological definition. A G-protein-coupled receptor, often abbreviated as GPCR, is a type of metabotropic receptor. Unlike simple ion channels that just open a pore, metabotropic receptors initiate complex metabolic steps. A defining physical characteristic of a GPCR is that it is a single long protein chain that weaves back and forth, spanning the cell membrane exactly 7 times. The outside portion of this structure acts as the docking station. When a specific neurotransmitter floating in the extracellular fluid binds to this outer part, it triggers the next crucial step in our communication chain. So, what happens exactly when that neurotransmitter binds to the outside? The binding causes the entire receptor to change its physical three-dimensional shape. This structural shift is the key that unlocks the process inside the cell. The shape change allows the intracellular part of the receptor to bind to and activate a complex called a G protein, which is normally just resting attached to the inside of the cell membrane. Once activated, this G protein does not stay put. It actually splits apart into smaller subunits. These subunits then travel along the inside of the membrane, acting like the bank workers who receive the walkie-talkie message, ready to carry out their specific tasks. Following the journey of those traveling G-protein subunits, we reach the next stage of the process. These subunits move along the membrane until they encounter specific targets known as effector proteins, which they can either activate or inhibit. These effectors are often enzymes, whose job is to synthesize new molecules called second messengers inside the neuron. These second messengers are the true workhorses of the cell. They flood the internal environment, triggering a massive cascade of complex chemical reactions. This multi-step relay is exactly how a single neurotransmitter on the outside translates into widespread, profound changes deep inside the neuron. You might be wondering, why does the brain bother with this complex, multi-step process when it could just use simple ion channels that open immediately? The answer lies in 2 crucial concepts: signal amplification and prolonged modulation. While direct ion channels are great for fast, split-second reflexes, GPCRs are responsible for long-lasting changes in our brain states. They regulate our mood, our level of alertness, and our ability to form long-term memories. Without them, our nervous system would be entirely reactive, incapable of sustained emotional states or complex learning. Furthermore, because they control so many vital internal pathways, GPCRs are the primary targets for a vast majority of modern pharmaceutical drugs, including antidepressants and pain medications. To truly appreciate the power of a GPCR, we need to look at how it amplifies a signal mathematically. Because each step triggers multiple molecules in the next step, the signal grows exponentially. We can represent the total cellular response by multiplying the amplification factors at each stage of the biochemical cascade. In this equation, R total represents the total number of final target proteins that are altered inside the neuron. NG stands for the number of G proteins activated by a single receptor. NCAMP represents the number of second messenger molecules produced by each effector. NPKA is the number of protein kinases activated, and N-target is the number of final proteins each kinase modifies. If any of these variables increases, the final cellular response explodes in magnitude. Let us walk through a practical numerical example to see this amplification in action. Suppose a single molecule of norepinephrine binds to a GPCR. We want to find out how many final target proteins are phosphorylated. From laboratory measurements, we are given that one GPCR activates 10 G proteins. Each G protein produces 1,000 molecules of cyclic AMP. Each cyclic AMP activates one protein kinase A molecule, so that ratio is 1:1. Finally, each kinase phosphorylates 100 target proteins. We substitute these given values into our amplification formula, multiplying 10 by 1,000 by 1 by 100. The result is 1 million target proteins. This incredible physical result explains why GPCRs are so powerful. A tiny chemical whisper on the outside becomes a deafening roar of biochemical activity on the inside. Now that we have seen how massive the signal can become, we must ask, how does the neuron ever calm down? Another critical aspect of GPCRs is how long their effects last, which is determined by how quickly the G protein turns itself off. The active G protein subunit actually possesses a built-in internal timer. It slowly breaks down its attached energy molecule, GTP, into a depleted form called GDP. This chemical conversion inactivates the G protein, stopping the cascade. Because this happens randomly to a large population of molecules over time, the number of active G proteins follows a classic exponential decay curve. Understanding this decay is essential for knowing how long a brain state, like alertness or pain relief, will actually persist. To model this internal timer, we use the exponential decay formula. This equation allows us to track the exact moment-to-moment decline of the signal inside the neuron. In this formula, G of t represents the number of active G proteins remaining at a given time t. G0 is our starting point, the initial burst of active G proteins right after the receptor is stimulated. The constant E is the base of the natural logarithm, approximately 2.718. The variable K is the decay constant, specific to that G protein, which tells us the fraction of proteins that turn off every second. Finally, T is the time elapsed in seconds. As time t increases, the negative exponent grows, causing the total number of active proteins to drop off smoothly. Let us apply this decay formula to a specific scenario. We are given that a neuron has an initial burst of 500 active G proteins. The decay constant for this specific protein is 0.1 per second. We want to find out how many G proteins are still active after 10 seconds have passed. First, we plug our values into the formula. We simplify the exponent by multiplying negative zero point 0.1 by 10, which gives us negative 1. The value of e to the power of negative 1 is approximately 0.368. Multiplying our initial 500 by this decimal fraction gives us our final answer, 184. Physically, this tells us that even after 10 full seconds, an absolute eternity in the nervous system, more than a third of the proteins are still actively changing the cell's chemistry. Let us bring all these concepts together by looking at this complete schematic of the signal transduction pathway. At the top, you can see the extracellular fluid, where a small neurotransmitter molecule is binding to the GPCR. Notice how the receptor weaves through the lipid bilayer exactly 7 times. Down below, on the intracellular side, we see the G protein complex. You can clearly see the alpha subunits separating from the beta and gamma subunits after activation. Follow the arrow as the alpha subunit moves along the inner membrane to dock with the large effector enzyme, adenylyl cyclase. Finally, look at the bottom right, where the enzyme is churning out a cluster of cyclic AMP molecules. This diagram beautifully captures the moment the outside signal crosses over to become a massive internal cascade. We have covered a lot of ground today, so let us review the most important takeaways. First, G protein-coupled receptors are the brain's master modulators. They do not just open doors, they translate extracellular chemical messages into massive intracellular biochemical cascades. Second, by utilizing a middleman, the G protein, these receptors achieve incredible signal amplification, turning a single molecule into a million modified proteins. Finally, because these G proteins have internal timers, they create prolonged cellular responses. This is what allows our brains to maintain sustained moods, form long-term memories, and respond to life-saving medications. You now understand the foundational mechanism of how the brain regulates its own complex behavior. Great job, and keep exploring the wonders of neuroscience.